This video is for the Biology Keystone in Pennsylvania. This time, we are reviewing Anchor 5, Cell Growth and Reproduction. This anchor requires you to describe the three stages of the cell cycle. That includes interphase, nuclear division, mitosis or meiosis, and then cytokinesis. And then we need to explain how genetic information is inherited. But let's start with the cell cycle. Most of the time in a cell's life, it is spent in interphase, just doing the normal regular cell processes. Interphase is divided into three subcategories. G1 is just time for the cell to grow. S1 stands for the synthesis phase. In this case, that's where DNA is being replicated. G2, the cell grows some more and then enters mitosis. Mitosis is the division of the nuclear material, that is the chromosomes, and then cytokinesis, the dividing up of all the organelles. You'll notice on this diagram that there are some stoplights. Those stoplights represent checkpoints, where if the cell hasn't grown enough, it doesn't make it through that checkpoint, and as a result, the cell has to continue to grow before it can enter the next phase. At the end of G2, there's another checkpoint to make sure all the DNA is replicated appropriately, and an anaphase is another checkpoint to make sure all the chromosomes are being divided evenly. If not, sometimes the cell will stay in that phase, sometimes the cell will just program to die, apoptosis. The idea of mitosis is how do you divide up the chromosomes evenly? So if a human body cell has 46 chromosomes, we need to make sure that after those chromosomes are replicated, that they're divided evenly between the two new cells. That process is called mitosis. And the reason that it's important is because chromosomes consist of coiled DNA. DNA just coiled up around histone proteins and organized in the chromosomes. So just to kind of refresh your mind, the parts of DNA consists of this phosphate, sugar backbone, and then some nitrogen base. So phosphate, sugars, and nitrogen base. Those nitrogen bases pair in a specific order. Cytosine pairs with guanine, adenine pairs with thymine. So in this little diagram, the orange represents a phosphate, the blue represents the sugar, deoxyribose, the green represents a nitrogen base, and the monomer is a nucleotide. This little diagram is just showing how DNA replication works. So during that S phase, DNA is going to be replicated, but how? Well, the twisted ladder double helix shape is unzipped by an enzyme called helicase. It splits the two strands of DNA apart. Then one of the strands is going to be copied by DNA polymerase, and it's going to be copied in the 5 to 3 prime direction, following helicase. The other strand is also going to be copied, but it's going to be copied in the 5 to 3 prime direction, called the lagging strand, the opposite way. So after the strand of DNA is copied, that is, in the strand, if there's an adenine, then the DNA polymerase will add a thymine. If there's a guanine, the DNA polymerase adds a cytosine. This is a picture of a bacteria DNA being replicated. So remember that bacteria DNA is not in a nucleus, but it does get replicated because bacteria divide by binary fission. They're just clones of one another. Remember the stages of mitosis with the acronym PMAT. If you want to remember the stages of the cell cycle, remember the acronym IPMAT, IPMAT. Interphase, prophase, metaphase, anaphase, telophase. Here we're going to start with prophase. In prophase, the chromosomes condense and the nuclear membranes begin to disappear. In metaphase, the chromosomes are lined up in the middle. Metaphase, middle. In anaphase, they're moving away. Anaphase, away. And in telophase, you have two new nuclei. The way mitosis works is it's just making a clone of the original cell. So if there are 3 chromosomes in the original cell, there's going to be 3 chromosomes in the daughter cells. Spindle fibers attach to the centrioles and make sure that those chromosomes are being pulled away to the opposite sides of the cell in equal numbers during anaphase. Finally, the last phase is cytokinesis, a division of all of the cell's organelles and cytoplasm. This is some pictures of plant cell mitosis. So you can see the cell, the first picture, the cell's in interphase, And then the next picture, the chromosomes are condensing. That's prophase. Metaphase, they line up in the middle. Anaphase, they start to go away to either side. And telophase, you have 2 new nuclei. Cytokinesis will be 2 new cells. One confusing thing about mitosis and meiosis is the idea of chromosomes. This is a picture of a chromosome. The center of the chromosome is a centromere. Whenever we count chromosomes and we say things like humans have 46 chromosomes, we're counting the number of centromeres. Whenever that chromosome is duplicated during that replication time, we call each side of that chromosome a chromatid or a sister chromosome. So if you look at the picture on the left, there are 12 chromosomes in that picture. In the picture on the right, there are also 12 chromosomes in that picture. The difference is the picture on the right has 24 chromatid. During cell division, then those daughter cells are going to wind up with the correct number of chromatid, but in that point, there'll be chromosomes. Meiosis is the division of sex cells, gametes. Mitosis is the division of body cells, skin cells, things like that, bone cells. When you're looking at meiosis, notice some significant differences because meiosis is not going to work the same way as mitosis. In the case of meiosis, during prophase, instead of having chromosomes, we have homologous chromosomes. That is, there's a set of chromosomes together. It's during that time that sometimes pieces of those chromosomes will cross and swap places. This increases some genetic variation, but that process is called crossing over. Meiosis part 1 means that these homologous chromosomes are paired up together. And during metaphase, they line up in the middle. Anaphase, they move away to opposite poles. And then we get 2 new cells. What's a homologous chromosome? Well, a homologous chromosome is a chromosome that has the same size, same shape, same genes. Well, if they have the same genes, why bother crossing over? Well, that's a good question. Crossing over is this idea of increasing genetic variation. Well, just because the chromosomes have the same genes doesn't mean they have the same variation of the gene. For example, if the gene is dimples, you might have the dimple variation, or you might have the no dimple variation. In that case, the crossing over can swap some of those genes. Now notice, during crossing over, it's more likely that the genes at the end of the chromosome will cross over, and it's less likely that genes that are close to the centromere will cross over. After the cell goes through prophase, metaphase, anaphase, telophase part 1, it's going to go back through those phases again in meiosis part 2. No interphase, just prophase 2, metaphase 2, anaphase 2, and then telophase 2. Notice in metaphase 2, we're starting out not with homologous chromosomes, but we're just starting out with a complete chromosome, 2 sister chromatid together. In metaphase, they line up together. In anaphase, the chromatid are now separating and moving to opposite poles. So that the 4 new gametes, the 4 new sex cells, the 4 new sperm, the 4 new eggs, will each have half the number of chromosomes that we started with. So in meiosis, we start with 46, replicate in order to get 92. Meiosis part 1, we wind up with 46. And then meiosis part 2, we're going to wind up with 23 chromosomes in each cell. So when you're looking at diagrams for meiosis part 2, look for chromatid, like sister chromatid together, and look for pictures that have 2 cells dividing instead of just 1. What if the chromosomes don't separate correctly? Well, that's called non-disjunction. It's sometimes where meiosis goes wrong. If mitosis goes wrong, we call it cancer. If meiosis goes wrong, it's going to be a chromosomal disorder that we pass on to our offspring. If the non-disjunction happens during meiosis part 1, notice that all 4 gametes are affected. 2 of them have too many chromosomes, 2 have too few. If the non-disjunction happens during meiosis 2, notice that only half of the gametes are affected. There are some significant differences between mitosis and meiosis. Mitosis, body cells. Meiosis, sex cells or gametes. Mitosis is going to end up with the same number of chromosomes we started with. So if we started with 46, we're going to wind up with 46. Meiosis is going to end up with half the total number of chromosomes we started with. If we start with 46, we're going to wind up with 23. That 23 number is called the haploid number, the half number. The diploid number is called the double number. That's the complete number. So in meiosis, there's one replication but two cell divisions. In mitosis, there's one replication and one cell division. In mitosis, we wind up with two daughter cells. In meiosis, we wind up with four daughter cells. So they're completely different processes to divide the nuclear material, the chromosomes, in body cells as opposed to sex cells. I hope that helps you understand biology, Keystone anchor number 5.